Morning students, today we are going to start our next chapter. Name of the chapter is cell cycle and cell division. So cell cycle and cell division is a cyclic process, and it contains the two phases, that is interphase and dividing phase. Interphase is known as resting phase. In this phase the cell is metabolic active, but the cell is non-dividing in that phase, in interphase. That's why it is known as resting phase. Interphase contain three substages, that is G1 first growth phase, S is synthetic phase, and G2 is the second growth phase. Once G1, S and G2 is completed, then the cell will enter into the dividing phase known as M phase. So in G1 phase, the first growth phase, protein synthesis is there, and in S phase chromosome duplication is there, and in G2 phase the RNA and the protein, remaining RNA protein synthesis will take place. And after the G2 phase, then the cell will enter into the M phase, which is the actual phase of cell division. So we can see the cell cycle contain two phases, that is interphase and dividing phase. And interphase G1, S and G2 is there, and in dividing phase the division of cell will take place in the two substages, in which one contain the division of nucleus known as karyokinesis, and then the division of cytoplasm is there known as cytokinesis. So again we can see here G1, S and G2, and then it will enter into the main dividing phase, in which first there is division of nucleus, and then it will enter into the division of cytoplasm. And once the actual division is completed, then cell again may enter into the G1, S and G2 phase. Now the division is of two types, the mitosis and the meiosis. First we start with the mitosis. Mitosis is the process of cell division in which there is production of two daughter cells from a single parent cell. The daughter cells are identical to one another and to the original parent cell also. So mitosis can be divided into four substages, that is prophase, metaphase, anaphase and telophase. After telophase, the cell will enter into the cytokinesis step. Interphase. Interphase is the phase in which cell prepare for the division. In interphase, we can differentiate the interphase for animal and plant cell here in this slide. In animal cell, in interphase, DNA replication is there, organelle replication is there, cell increase in size. In plant cell, DNA replication also take place in interphase, organelles replication take place, and cell increase in size here. So we can see this is the actual slide of interphase in animal cell and plant cell both. Then the cell will enter into the actual division phase, and first is here the division of nucleus will take place. So prophase. In prophase, packaging of DNA will there, means condensation of DNA will take place to appear to form a chromosome. We can see with this picture also here. So DNA condensed to form chromatid and chromosome will appear in this prophase. And in prophase, the nuclear membrane disintegrate or degenerate, we can say the disappearance of nuclear membrane will take place. These are the two actual slides of animal cell and plant cell. Then metaphase. In metaphase, the cell prepare chromosome for the division, and in animal cell and plant cell both, chromosome will line up at the equator position or at the center of the cell, and spindle fiber will attach to these two chromosomes with opposite poles. Yes, we can see here the chromosome in animal cell and in plant cell. These chromosomes are present at the center of the cell, having the spindle fiber. These are the actual slides. Next is anaphase. In anaphase, the chromosome will divide. So spindle fiber pulls the chromosome, and then chromosome will split into half containing the chromatids, and these will move towards the opposite poles. Yes, we can see here the chromatids of chromosome separate in anaphase, in animal cell and plant cell. Next is telophase. In telophase, the division of cytoplasm will initiate, and as the chromatid reaches the two opposite pole, the DNA spreads out and the two nuclei will form, and cell wall will pinches in to form the two new daughter cell. We can see in this slide This is the animal cells, and these are the plant cells. Next, plant mitosis. We can see in the interphase, these are the slides of interphase first, then metaphase. Chromosome will present at the equator. In anaphase, separation will be there, and in telophase, the cytogenesis will take place. Now next is meiosis. Meiosis is the type of cell division by which germ cells are produced. One parent cell produces 4 daughter cells, and in meiosis, reductional division is there, means the chromosome number will reduce to half. But in mitosis, the chromosome number will remains the same. So during meiosis, DNA replicate once, but the nucleus divide twice. And meiosis also contain 4 stages. And meiosis 1 contain prophase 1, metaphase 1, anaphase 1 and telophase 1. In prophase 1, each chromosome duplicate and remain closely associated. These are called as sister chromatid. Metaphase 1, chromosome will present at the equator or center. Anaphase, chromosome pair separate, and in telophase, 2 daughter cells are formed with each daughter cell containing the 1 chromosome. You can see in prophase 1, early prophase, we can see here nuclear envelope. Middle prophase, chromosome condense. Middle prophase, we can see the chromosome condense to form, we can say DNA condense to form a complete chromosome, and X-shaped structure is formed known as chiasmata. And in late prophase, the nuclear membrane disintegrate, and the shortening of chromosome continue. In metaphase, chromosome will present at equatorial area or central area to form equatorial plate. In anaphase, the chromosome will separate, and in telophase, there is division of cytoplasm will take place. Now meiosis 2. Meiosis 2 also contain prophase 2, metaphase 2, anaphase 2 and telophase 2, and this meiosis 2 is similar to that of mitosis. In prophase 2, DNA does not duplicate. Metaphase 2, chromosome will present at the center. Anaphase, the separation of chromatid will take place, and in telophase, cell division is completed. We can see here prophase 2, the chromosome condense again, and following a brief interphase in which DNA does not replicate. In metaphase, chromosome will present at the center. In anaphase, chromatid will separate, and in telophase 2, 4 cells are formed. So meiosis form the 4 haploid cells. We can see from this slide also. Meiosis 1 will produce 2 cells, and meiosis 2 will again divide to form total 4 cells from 2 daughter nuclei. So now the difference between mitosis and meiosis. Mitosis will take place asexually. In meiosis, sexual reproduction is there, and mitosis cell divide once, meiosis cell divides 2. 2 daughter cell will form in mitosis, but 4 haploid daughter cell form in meiosis. Genetic information is identical, and meiosis genetic information is different. So this is all about mitosis and meiosis.